anatomists and physiologists. This video is all about blood vessels. We're still in cardiovascular system. We're just jumping into a new chapter, chapter 19. So let's get started. Our goals for this lecture, we have a couple of videos in this series, but our goals are to talk about blood vessel anatomy, how generally are blood vessels structured. We'll talk about the walls of the vessel, arteries versus veins versus capillaries. What are those things? And then we'll also learn about, mostly in the lab, the major vessels, those main arteries and veins of our body. Then we'll jump into the physiology, what goes into all of that. And then lastly, we'll talk about how blood is circulated and that path that blood takes through the body. And all of these will help us in our understanding of the cardiovascular system at large. So since part one of this video series is about anatomy, we first need to think about what do blood vessels even do? So what are they doing all day? And they're part of the cardiovascular system, right? So they're doing transport, just like the heart. Transport of what? Transport of blood. Blood has cells and nutrients and gases like carbon dioxide and oxygen. We move all that stuff around. And if we think back to our cardio lecture, we did a drawing about how vessels are organized in the body. So if we have a heart, let's draw a heart. So we have a heart, and then we have vessels leaving the heart. So we have vessels leaving the heart. Those vessels leaving the heart, those are arteries. And then we have vessels coming back to the heart. Those vessels are called veins. I should have moved this drawing over, so I'm going to do that. So generally we have arteries are those vessels leaving the heart and veins are the ones coming back. But in the middle there's stuff. And we drew this in our cardio lecture. So first I'm going to draw a big vessel. And then that vessel will get smaller. And then that vessel will get big again. And then I'll do the same on the other side. Big vessel, and then it'll get really small, and then it'll get bigger. So generally how we label these is that arteries, those are those big vessels leaving the heart. And they leave the heart. But then arteries, they branch out. Every time I hear arteries, I'm always thinking branch. So arteries, they branch out and they get so small. They get a lot smaller that it's kind of silly to call them arteries anymore. So we call them little arteries, arterioles. Arterioles. And then those arterioles get so small, it's silly to call it an arteriole because just one layer of cells can fit through. This teeny tiny vessel is called a capillary. That's where gas exchange happens. We drop off oxygen, pick up carbon dioxide. Capillaries often exist in capillary beds, and I'll show you some pictures of what that might look like. And then capillaries, they combine in on themselves until they're bigger again, and then it gets silly to call them a capillary, so we call it a little vein, a venule. Venule. I'll actually do this in blue. Instead of venule in red, I'll write venule in blue. We call it a venule, a little vein, and then those venules combine and they merge together and they combine some more. So whenever I think arteries, I'm thinking branching, and arterioles, I'm thinking branching. But whenever I'm thinking venules and then the next one, which is veins, and then of course back to the heart, whenever I'm thinking venules and veins, I'm thinking combining together. Because all those venules combine into all those veins, and those veins combine into the SVC and the IVC mostly when we go back to the heart. Okay, so I know that I've drawn things in red and blue to denote arteries and veins, but what I really mean about red and blue, we talked about it before, is that red is indicating oxygen-rich blood, and blue is indicating oxygen-poor blood. Most days, in systemic circulation, it is true that arteries are red, they're carrying oxygen-rich blood, and veins would be considered blue in our textbook, oxygen-poor blood. In systemic circulation, I think that's true, but in pulmonary circulation, we take oxygen-poor blood away from the heart and arteries, and we return oxygen-rich blood in veins. So in pulmonary circulation, it does get a little backwards. So just keep that in your brain. Okay, so blood vessels, that's what they do, transport. If we were to take a cross-section of an artery and a vein, this is what we would see, this picture up here. Let me zoom in on it. So we'd see an artery is really circular and it has this like really thick purple layer. And then a vein is kind of squished and that purple layer is a lot thinner. So that's what we're looking at here. Okay, so it's in cross-section and these top layers also cross-section. Okay. So what's going on the left side is we have an artery and then the right side we have a vein, and then at the bottom here we have a capillary. Remember I told you about capillary beds, right? So we have a capillary bed there in the middle, and I'm going to zoom in on it. You see how you could like lay down there like a little hammock? Yeah, a capillary bed. It's just a bunch of interconnecting teeny vessels. Okay, so back to arteries and veins. So the main difference between an artery and a vein is its structure, because structure leads to function, right? So I'm going to highlight some of the big things in yellow. So arteries and veins have three major layers. There's an inside layer, a medium layer or middle layer, and an outside layer. So we have an inside layer, a medium layer or a middle layer, and an outside layer. So every time that I see these words, I want you to think about, or new words, I guess, you should think about what that word really means. And I'll give you some conventions that we can take with us. So let's see, I'll do my annotations in pink. So the first layer is called the tunica intima. And so just so you know, tunica, that word tunica, in anatomy and physiology land, it means layer. Or you might hear it called tunic. So that word tunica means layer. So this is the intimate layer. Tunica intima is the innermost or deepest layer of both an artery and a vein. And what that tunica intima consists of is a bunch of endothelium. Endothelium we've learned about before. I told you it would come back, and it did. It's simple squamous epithelium. And then there's like some connective tissue to lick and stick it all together. Okay, there's an endothelial layer and an elastic membrane, which is just connective tissue. Okay, there's an inside layer, tunica intima, the intimate layer, because it's like intimate with the blood. Speaking of this section on the inside, like if blood were to pass through there, that hole is called the lumen. It's at the bottom here, but I don't know on the video if you'll be able to see it, so I'll label it here for you. So this inside layer is called the lumen. Lumen. The lumen is usually the hollow inside part of an organ. So if an organ has a hollow inside part, your guess is that it's called the lumen. That'll come back, okay? Okay, so we have the inside layer, the deepest layer, the tunica intima, endothelium and stuff. The next layer is the middle layer, the tunica media. And the tunica media is a muscle layer. So always remember, tunica media is for middle, but the M in media is for muscle. And what kind of muscle is that? It's not skeletal muscle, because that's with the bones. It's not cardiac muscle, because that's with the heart. So it's smooth muscle. Smooth muscle, we can abbreviate like this: big S, little m, big M, smooth muscle. So smooth muscle is not striated, so it doesn't have that actin-myosin sliding filament situation going on. But it's also not voluntary, so we don't have a voluntary control over it. And vessels, they can get bigger, they can get smaller. That's called vasodilation and vasoconstriction. That's done by the tunica media, okay, the muscle layer. And then there's like some elastic fibers in there, and elastic fibers in there are for the stretchiness. We stretch and we come back, okay? So what I want you to see here is that in arteries, that tunica media layer is a lot thicker compared to veins, a lot thinner. And we saw that when we did our heart dissection. We saw that the arteries, the aorta, the pulmonary trunk, were a lot springier, they had a lot more substance to it compared to the veins because, in large part, because of this tunica media layer. Okay, and then the last layer I want to talk to you about is the tunica externa, the external layer. I'm breaking down this word, every word that I'm teaching you, because I think you should too whenever you're in your notes. Write down that word. Okay, so the external layer is the superficial most layer. It's a lot of fibers and stuff for structure, okay? So the tunica externa is the outermost layer. And that outermost layer, the tunica externa, is mostly like collagen. And we know that collagen is really good for fibers, or really good for structure, rather. But sometimes our vessels are really big, okay? So how these vessels get, and all of the cells of these vessels get their nutrients is from the blood that lives in the lumen, okay? But sometimes our vessels are so big that we can't let all the nutrients pass to the outermost layer. So sometimes the tunica externa in a really big vessel has its own vasculature, and I'll write that here. I'll actually highlight it for you in pink. So sometimes our vessels get so thick and so big that the external layer, the tunica externa, actually needs its own blood supply. That has its own name. It's called vasa vasorum. Vasa vasorum. It's a special name for the blood vessels of the blood vessels supplying to the cells. It's very meta, but remember that the heart had its own vasculature. Sometimes blood vessels do too if they're too big. It's really meta. Okay. So next things next. I'm going to show you some differences, some different stuff that we got going on. This is table 19.1 from your text. And this is all of the different bits of blood vessel anatomy. First, I'm going to tell you what's going on with this table, and then I'm just going to show you some pictures. Okay, so there's different types of vessels. There's arteries, and there's veins, and there's capillaries, but there's different flavors of those, okay? And then this part, the next column, is the average lumen diameter. So that means like how big is that part that blood flows through? How big is it? And how thick is that wall? Is it thick with one C or thick with many C's, okay? And then lastly, what kind of tissue makes it up, okay? So smooth muscle is the dark red here. We have endothelium, which is pretty much the same across the board. And then like how much fibrous or collagenous tissues, that's the white. Just if you care to know. Okay, so let's jump in. The first thing I want to show you is, first we have arteries and different flavors of arteries, and then we have some capillaries. We have different flavors of capillaries, but that's on its own slide. Then we have some veins, and I'll talk to you about those. Okay, so let me zoom in on the arteries so we can see. Okay, so in arteries, we have a lot going on, to be honest with you. In arteries, we have arteries that need to be really, really good at being springy, at withstanding pressure. And those are those vessels that are really close to the heart. And those are what we call elastic arteries. I'm going to highlight that one as I talk about it. So elastic arteries, that's these ones, elastic arteries. Elastic arteries have lots of springiness to them, meaning that for the example, aorta, as that ventricle pumps that blood out, there's lots of pressure. So we need to expand, and then we need to come right back. So these vessels are often located really close to the heart, like the aorta and all the branches off of the aorta. And notice that the lumen here is really wide, so that diameter of the lumen of the blood vessel can be really big. And it can be really stretchy and very elastic arteries, okay? So these vessels are really good at handling that pressure because they can expand and contract because they're elastic. But if that elasticity is lost, for example, in atherosclerosis, when that vessel kind of hardens, we can't do that expand and contract thing very well. Okay, so the next one is muscular arteries, and I'll circle those in pink. Muscular arteries, are more muscular. They have lots more smooth muscle, and these are more distal to the heart. So I would call elastic arteries proximal to the heart, and I would call muscular arteries distal to the heart, if we're comparing the two, okay? If we're comparing those two. And the muscular arteries, they tend to be the arteries that carry blood to specific organs, like to the stomach, to the arm, to the leg, that kind of thing. And so most of the arteries that we'll learn in class are actually muscular arteries, okay? But they're really good at vasoconstriction and vasodilation because they have less of that springiness, they have more of that muscliness. Okay, and then we have a little arterial. See how small it gets? Anyways, we also have some capillaries. There's different flavors of those. I'll show you on the next slide. But then I want to spend some time talking about veins, venules and veins. Venules, little veins, veins, regular veins. So veins, they're the same as arteries in their structure in that they have those 3 layers, but those 3 layers are a little bit different. Notice that lumen is quite large. That tunica externa, quite thick in comparison, okay? And that's because we need a little bit of structure, and also because of this diameter that it's bigger. It can hold more blood. So most of our blood at any one time is in our veins, most likely, okay? Veins, because they have a bigger diameter, notice this is still coming back, veins have a bigger diameter, so they usually have less pressure in addition to that. Because they have less pressure, sometimes veins need help in getting blood back to the heart, and so veins have valves. So I'll scooch you over this. Have valves. And we learned in the heart unit that valves prevent backflow. So as that blood is going back to the heart, if that blood were to lose pressure and start to come back down, that valve would prevent it until in the next heartbeat will then propel that blood upwards. So that prevents backflow. We have several different flavors of veins, but really one of the ones that I want to talk to you about are called venous sinuses. So there's a flavor of vein and it's called venous sinuses, and I'll write this to this side. Example, a specific example, venous sinuses. Venous sinuses are veins. They're usually pretty big, and their walls are pretty thin. They're pretty big, but their walls are thin, and they're usually supported by the structures around them. So we've learned about the coronary sinus that brings blood back to the heart from the heart, okay? But we also learned in 430 about the dural venous sinuses in the brain that help bring all of the excess fluid from CSF back into the blood, okay? So those are usually supported by surrounding structures. But a homeostatic imbalance of veins, like a clinical application, would be varicose veins. Varicose veins. And varicose veins, they can happen because of a lot of reasons, but they're really, really common in things in folks who are overweight, in pregnancy, or they can just be hereditary. Varicose veins are when especially superficial veins of the lower limbs kind of get a little bit dilated and they get kind of wonky. And when they get a little bit dilated and a little bit wonky, they can't really drain that well. So venous drainage is not so great. So when those walls stretch, venous return goes down, and then we kind of get this like pooling of blood in that region, and it can start to be painful if that gets kind of big, or if it's in like a really uncomfortable area, like on the back of the knee, that kind of thing. But sometimes this happens when venous pressure is elevated. So when the pressure in that vein gets really high, like in a bowel movement, like in delivering a baby, that kind of thing, that's when you get varicose veins in the anal vessels, and we would call that a hemorrhoid. Not so comfy. Okay, so we talked about arteries, we talked about veins, and kind of the different flavors of each. But also I want to talk about damage. If vessels get damaged by like a cut, or if vessels are unable to pass blood through that region, we might want an alternative route. Just like if the freeway exit was closed, you'd want to be able to circle back and take the back roads. That's called an anastomosis, plural anastomoses. These are alternative routes of blood flow. So if you can't go route A, you can go route B and still supply blood to that region. In arteries, when we have this happen in arteries, this is called collateral channels, basically just meaning that we have lots of ways to get to the same region. This is really common in things like the brain, because we want to make sure that we're supplying blood to our brain, because our brain is kind of important. But also to our joints, because our joints are always moving. If I bend my elbow, I still want blood flow going to all the things, but if I pinch those vessels, I want more blood flow still happening, regardless of if I'm pinching a little bit of blood vessels there. It's also really common in abdominal organs and the heart, all these things to make sure blood gets to where it needs to go. But it's not the end-all be-all in arteries. This is not a common thing in arteries. It's more like a special thing in arteries. So if arteries get damaged, then that can start to lead to circulation problems. But in veins, this is super common. Veins anastomose or combine and may alternate routes all the time. Your textbook mentions that you should take a look at the back of your hand or any region of your body that you can see kind of your veins, and you can really see that your veins kind of take their time and get where they need to go. They have lots of different types of anastomoses that go on. So if veins get obstructed, because there are so many anastomoses, alternative routes of blood flow, it tends not to be a big problem. Okay, so now I'm jumping into capillaries. This is a picture from your textbook. I'm going to zoom in on each of these. So there's three main types of capillaries I want to talk to you about. So the first type is a continuous capillary. Continuous capillaries are the most common capillaries that we have. And when we think about a capillary, before I jump into the specific types, I should draw for you what we know. So capillaries look different, but generally this is what happens. So we have a capillary here, and if I were to draw a blood cell, a red blood cell, it would kind of like only fit one cell deep, or one cell at a time, single file. So here's my little red blood cells, kind of look like deflated jelly donuts to me. So it's really just like single file cells, okay? So that's what a capillary kind of looks like. It's very, very small, one cell, and those cells kind of like stacked together, almost like pennies. Okay, so now that we're here. So continuous capillaries. Continuous capillaries are the most common types we got. And I want you to look at how those cells are joined together. There's little gaps there, but they're joined by tight junctions. They're joined by tight junctions. So what that means is we do have little spots. Those little spots, or little holes rather, are called intercellular clefts that allow substances to pass through. But most of those cells are joined by tight junctions. We also have these little, in yellow, these little spidery shaped cells called pericytes. Peri meaning around, cyte meaning cell. So around the cell, around the capillary cell. And so these pericytes, they help with permeability. They help make sure that those capillaries can repair themselves. They have a little bit of a stem cell capacity there. And so it's really important to have capillaries in the areas you need and how you need them. And what I mean by that is continuous capillaries are most commonly found in our body because, well, We don't always need tons and tons of exchange. Versus in a fenestrated capillary, notice how different that looks. I'm going to zoom out so you can compare. So look at those junctions. We have little teeny tiny spots in tight junctions. But in fenestrated capillaries, look at how many holes there are. Those little holes are called fenestrations. It kind of reminds me of Swiss cheese. But we find fenestrated capillaries in areas of high exchange. So either lots, oops, I'll shrink my marker here. Lots of filtration. Filtration happens when stuff leaves the blood, or we have lots of absorption. Absorption is that fluid going back into the blood. So this happens in like our kidneys, where we do lots of filtration and absorption, but also in our small intestine. That's where we do a lot of our digestion, right? So we need to absorb a lot of those nutrients from our food, and also in the endocrine system. That's where we release a lot of hormones. We need to release stuff into the blood. And so having these holes in the capillaries becomes very, very convenient because stuff can more freely flow through. But still, we have some tight junctions going on here, but Swiss cheese at the same time. Okay, the last flavor or the last type of capillary that we have are called sinusoid. Sinusoidal capillaries. Sinusoid is kind of like an S-like shape. So these capillaries kind of like meander, they take their time. These sinusoid capillaries are very, very permeable. Sinusoid capillaries are super permeable. Still, our cells are kind of joined together by like tight junctions, I guess. But even the basement membrane is not complete. That's just the outer clear part, okay? That outer membrane holding everything together is not even a real membrane around because it's kind of broken up here. So this is in areas of high exchange. So we find this in like bone marrow. That's where we make blood cells. So we need to release those cells into the bloodstream. In the spleen, where we get rid of old blood cells. So we're having cells cross that border. But also in the adrenal medulla. So your kidney kind of looks like this, right? So your kidney is like a little kidney bean. And then your urethra comes off of it. But if your kidney was wearing a party hat, like that, that's the adrenal gland. We'll get to it. But anyway, it does a lot of hormone stuff. We'll get to it. Okay, so that's sinusoid capillary. So it does a lot of stuff. And blood, because there's so much exchange, really slowly flows through it. So I kind of think of these as like meandering, taking their time capillaries. Alrighty. So moving on from the types of capillaries, capillary beds are not always needed. If we're not actively digesting our cheeseburger, we might not actively want a ton of blood flow to our stomach and all of our GI tract. If we're not actively doing that. And so we can kind of turn off that capillary bed where there's not as much blood flow. So notice that we have an arteriole. The arteriole flows into a capillary bed, bunch of gas exchange and stuff happening here. Then we flow into a venule, and then we leave, head back to the heart. So if we don't want that to happen, then we'll just constrict this little portion right here. That's called that little portion is called the terminal arteriole. So that terminal arteriole will vasoconstrict, get smaller, and then blood will be restricted through that area. It's kind of nice if we don't want blood to go there. This happens in most regions of the body, but some regions of the body are special, and one of those regions is called the mesenteries. The mesentery. The mesentery is a bunch of serous membranes around the intestines, and so the intestines are really important for digestion and that kind of thing. And so what happens is, instead of just shutting off that whole capillary bed, we still maintain blood flow through there. We still maintain blood flow, but how these gray areas are shut off is because we have what's called precapillary sphincters. A sphincter is something that can close and open. Usually it's closed. And so that sphincter will close. It's kind of like a muscle. kind of restrict that blood flow through the rest of the capillary bed. So it's not really happening there. But that road that stays open through there, that is called a meta-arteriole. Meta-arteriole is the one that keeps blood flow moving from A, arteriole, to B, the venule. But we just shut off all the extra stuff. And that's only in the serous membranes of the intestines, the mesenteries. Okay, that's where I'm going to end this video. Next time we'll pick up on part 2, circulation physiology.